I thought it was.
I thought you actually checked when you told me.
I did.
I might have found out last night. That's all good.
So it's still 5 minutes.
I mean, I can give them 5 minutes out too. So the title is Balancing Complexity and Practicality in Vitro Models for Tox Testing and Precision Medicine. I have a conflict of interest. I own equity in a company that develops solutions for high throughput tox testing and multiculture applications, and we do show some of that technology in here. So take that with a grain of salt. So balancing complexity and practicality. So how do we really think about this and complexity and practicality? The way I think about it is they're sort of competing ideas, right? That the more complex you have, sort of the least practical that we can implement these things, the least, it's much more difficult to run them. There's increased costs, increased time, increased operational complexity, things like that. So these are sort of in conflict. And then in every model, we always think about how relevant these are to humans. And in tox, we're actually very interested in also protecting fish and wildlife and things like that. So this isn't always human relevance, but that's where the money is. And I think we find out there, you know, actually going in different species and things like that, there's a much broader application approach to this that you maybe don't always just want to think about humans, right? We want to sort of save the world too, if we can. So if we map out where people would be, sort of the most relevant people, and also sort of the most complex would be these two outstanding scientists here on the model. In the toxicology, typically what we've done for the last 50 years is basically done a lot of rodent studies and tried to use those to predict what's going to happen in the human. And so if you think about a mouse where it is, it's really just as complex as a human. It's just as operationally complex. If you read Covance's human studies subject thing, and you read a lot of our mouse animal cage things, we each house 4 animals in a room. Covance houses 4 animals in a room with their people. So there are actually a lot of operationally complex. They're relatively similar, right? The biology, we can't really tell the fine details of how different they are. So really they're on the kind of same level of complexity, but less human relevance, right? The problem that we have in tox and why this is failing is that we've used this model, this approach, and we've been able to study about 2,500 chemicals, and most of those, really only 200 or 300, in sort of where we feel really comfortable doing extrapolations and things like that. But we have 80,000 chemicals in commerce. This number's in a little bit of debate, but then there's far more chemicals and sort of drug potential things around. But for this reason, the National Academy basically said that we need to shift from this strategy where we simply do mouse studies to predict human disease to this prioritization approach. And that basically, the underlying hypothesis assumption is that we know basically that there's 30 or 40 ways that things can kill you, that things can really screw up biology. If we think about those 2,500 studies that we've done in different chemicals, we can map them back to a select number of pathways. So if we understand those pathways and we build sort of monoculture or very simple assays to flag things that might act in those ways, then we can prioritize, right? So we can screen these 80,000 in these much simpler models. We can use that to then select the really nasty looking chemicals, dose them in animals, and determine what the real risk is to human health. So this approach is what was adopted in 2007. I will say that, so I was just at the Society of Toxicology meeting. This was what the National Academy said we need to do. Everybody instead is trying to skip and just do this. And I think there's a lot of very fundamental problems with that, because now they sit and they're just crippled with the idea that, well, how do we actually assess risk from here? What do we do when we lose our model system? And the National Academy said, no, you should not lose your model system. We should prioritize and just test. But nobody's doing it. They're so frozen on developing models and risk parameters that will directly apply that they sort of crippled themselves. And it's a very, I don't know, the way I see it is a big problem in the field. But that's a little bit of an aside. I apologize for that. So if we think about, again, practicality, complexity, again, we said cost and time are down here, right? They're all in sort of conflict. But we really need to ask ourselves, what have we really lost by reducing that complexity? And then in our role here in the MMB lab, how can we use engineering to really make more relevant models? Because what we don't want to do is build a bunch of tools that are just as complex or more complex than the animal systems we have, because they serve no purpose for this context. We can use them for very cool stuff, to understand simple ideas in biology, to understand processes, to get under the hood. It's not like these things are useless in their own selves, but for this application, they basically are. There's no utility in building something that has just as complex, but much less human relevance. So what we need to do is make sure that we're sort of checking ourselves, building models that are more appropriate and more practical, so that really we can prevent the most disease with the limited resources we have. So I'm skipping over altogether, but I built this, a lot of this concept in writing, working with Kyung on the EPA HMAPS, or Cancer Map Center grant that we applied and were awarded for the EPA that Molly and Megan worked on. And from that approach, we identified a few really important ideas. That we use this Aoki framework, and I'll show you an example of it here in a little bit. And that's really, as Molly described it, I like that idea, it's just sort of a cheat sheet on how to build a model. It helps you identify the key things that pathologists, that stakeholders are used to seeing and maybe can use to actually assess risk. And it also is very important to include the microenvironment, that we know that these interactions are very important in regulating cell growth, proliferation, and toxicity. And by adding all that complexity, we saw that we had a lot of in vivo-like responses. We had tissue-level endpoints, which were good for building buy-in and increased sensitivity in those models. But there were some very specific things that I felt we could really improve on, right? That was a model that existed in the lab, and we applied to this concept. But if I was starting fresh, what would I want to do? What would I want to have if I could design it from the outset? Where would I start and what would I do? So some very simple ideas, right? That throughput was a very important thing. So with the Luminex system, if we're screening 80,000 chemicals, I could never get Molly to screen 1,000, even though I asked her 1,000 times, literally. There's a direct correlation there. PDMS is problematic, as we all know, because of chemical sequestration. And the model system as well. Breast cancer is not well studied. It has a long latency. We don't know a lot about how chemicals cause breast cancer, except through like activating estrogen receptor. And so there isn't a lot of real good parallels. We don't have those 2,500 animal studies. We don't have a bunch. Mice don't get breast cancer. They're actually a bad model for breast cancer. And so again, if you're thinking that you're going to do this and people are going to get buy-in, they're going to want that animal data. And I'm either going to try to generate it myself or I'm going to use what exists. And we're not a mouse lab, and it makes much more sense to use what exists. So if I think about what sort of model system we use, I looked around, and orofacial clefting is a very good one for a number of reasons that we'll go over. So that's sort of what we're going to talk about next. So as we were thinking again about getting rid of PDMS and what format to start in, I was at the Society of Lab Automation conference about a month ago, and you walk into the room and it's a huge conference center, right? And there are 40 robots this big by 30 different companies, all meant to interact with a microtiter plate. That's pretty much all they can do. They pick and squeeze about this far, and everything they do is microtiter based. The plate readers are microtiter based, the robots are microtiter based, anything you dispense in, the conveyors, the belts, is built to interact in this format. And so if you're going to compete, if you're going to make something useful for this space, you need to build it in this format. It's really the only answer. And so I developed this approach, sort of spun off of John's idea of micromilling being a very useful solution, to instead of just micromilling into flat plastic surfaces, to actually micromill devices directly into the bottom of weld plates. And so by doing that, basically, we can do pretty rapid prototyping. So CAD, 3 to 6 hours, and just to make simple changes can be 10 minutes, right? CNC milling, ½ hour to 2 hours, bonding, cleaning, sterilizing, about an hour. And then once we put things in here, we have days to weeks we've cultured these things. They can go back and forth in and out of the incubators. We can redose, do different things. And we found them to be very useful. And so this approach has been really enabling to develop new models. Bill Murphy gave the keynote at the Society of Toxicology meeting, which was last week, so 2,000 people in the room. And he put up the devices that we've made here, and actually directly called out a lot of the stuff that we've done, which was very nice in a room of 2,000 people. Stuck with plates now in developing the model. One of the reasons, many reasons to choose orofacial clefting, but of course we go to the biology. It's a very important disease, 1 in 700 live births, extensive morbidity, mortality. We can have cleft lip or cleft palate. But it's also a very good system to validate these models in because it's structurally simple, so it's not anything you need to build a huge crazy system on. Like I saw somebody giving a talk on the retina, and you talked about screening chemicals, and then he said, and we ran 2 chips, and so an N of 4 in each, and that was his screening, because it's, you know, he spoke the need for microfluidics and flow, and then tried to develop a retina, which is a 7-layered, you know, cell model, and very important, but, you know, it's not where maybe you want to start, is what I would say. A convergence of really critical embryonic pathways, Wnt signaling, hedgehog signaling, TGF-beta, AHR, really known classic toxicology things. Established genetic and chemical causes, so there's familial and there's chemical, and so we can study things like gene-environment interactions, which is really important. And then there's a lot of in silico data as well as a lot of animal testing data, and so we have this really rich resource to know what should happen. Okay, you know, we know what happens in the mouse, we know what likely should happen in the human, and in our model then, therefore. So there's a lot kind of going on here, but this is sort of what we set up our design parameters then based on this model. So we did the adverse outcome pathway for hedgehog signaling, and all this does is simply separate what happens on the pathway to a chemical coming into the body, binding and interacting with that first specific molecular interaction that starts to drive this, this progression. What cellular responses happen in response to that, what tissue changes occur, and then finally your adverse outcome. And so we have antagonism of the hedgehog signaling pathway in mesenchymal cells, and that leads to reduced proliferation, reduced tissue outgrowth, and basically you have 2 different sort of classic mechanisms with different molecular pathways that will block orofacial clefting. The first one being you have these 2 palatal shelves that sort of are like this. They elevate and then they grow out and grow together. The other one is that they grow together but don't ever fuse. So they can, you can either disrupt outgrowth so they can never reach each other to fuse, or you can disrupt fusion where those things will meet but they'll never fuse, and then they'll actually reverse and separate as the head grows during development. So we wanted to again match the biology. So it's a dense mesenchyme overlaid with epithelial cells. This hedgehog-directed outgrowth. A gradient is actually very important in hedgehog signaling. It's a morphogen, so it actually acts transcriptionally, drives different transcriptional signatures depending on its concentration. And we wanted to make it throughput compatible, so fluorescent and luminescent readouts. This is an overview of the hedgehog pathway, and we'll go look for it in a little bit too. But again, transcription of hedgehog ligand itself, it gets post-translationally modified with a cholesterol and a palmital group. Secreted into the extracellular matrix where it drives down, and so cells that are very adjacent to it see lots of ligand. Cells that are further away from this surface epithelium, this layer of epithelial cells, see less ligand. So it'll bind its receptor patched, which relieves repression of smooth end and activates this transcriptional pathway and drives classic target genes. So this adverse outcome pathway again helps identify those readouts that we want, and it helps us identify the cell types and organization, really to engineer a model that looks biologically appropriate. So to reverse this interaction, to reverse engineer it, we start with this 3D or epithelial mesenchymal organization. So it's outlined here. If we basically take the developing mouse, this is about embryonic day 14, I think, in the mouse. So basically now you take and you're sectioning the front of the nose off so you can see a cross section of the front of the palate. And you can see this really dense layer of mesenchymal tissue. So this is just structure, it's cells, organized in a very dense, and then overlaid with this red layer of epithelial cells. And so if you sort of draw a box, this is what we're trying to mimic. And so this is what that looks like. We have the dense mesenchyme embedded in an ECM. We overlay that with epithelial cells. And then we have two opposing each other because we'd like to get both outgrowth and fusion in the model. Although hedgehog is strictly pretty much proliferation and outgrowth, so that's what we're focusing on initially. Again, to design it, we want it to be throughput compatible. We want it to be easy to sort of understand and see, so we make it where we can image through the bottom and see these things. We can also then make it throughput compatible, right? So I have 40 of these devices in one plate. They can really start to run dose response curves, screen a lot of chemicals really, where we do this. And so here we have these microtissue wells, which are open to this top center well. There are phase guides that separate these two compartments, but they're open in the bottom. And you have pinning basically, so it's a little easier to see here. So this is the sort of underlying engineering that really makes the model work. If we have an empty plate, then we fill it with gel. What happens is actually these ports are open, but because of surface tension, the fluid will not, this liquid gel will not flow out. It'll polymerize in this pocket. And then we'll have this open face of gel that when we seed a cell suspension down here, we can coat that open gel face that's on the side. So this is sort of what it looks like. This is just adding gel. This is 1.75 microliters of gel with a few thousand mesenchymal cells in it. And it's pinned here, you can see, even though this channel is open to the side. And then now we've flowed, we've pipetted in these wells, and we're flowing fluid around this system. So you can see the cells sort of zipping along here. And because of laminar flow, again, you have flow in the center of the channel, but you don't have much flow on the walls. And that helps sort of keep, when you're flowing through, you can keep cells out of the center and over to the walls, where they'll selectively stick onto the side of that gel. So at this point, let's be honest with ourselves about what we really built. At this point, I would say it's actually almost just as complex of a system, and unless we find ways to sort of make this thing more useful and get more money out of it, just making one of these to do that is relatively complex. And so what are we actually going to get out of this? What did this increased complexity actually buy us? And what can we do to make it better? So if we just start to look at the biology that we get in this system, this is a cross-section of a developing orofacial prominence. And we can see this dense mesenchyme here, in vivo in the mouse, overlaid by the epithelium. And if we take a cross-section in the chip, we get the dense mesenchyme overlaid by the epithelium. So it's not perfect, but it's pretty good. Density is pretty good. Overlay is pretty good. And really, the biology looks correct. And a pathologist, won't look extra cross at you when they see this. So this is really cool, right? We've got something that really starts to recapitulate the biology. Again, we want to make this thing throughput compatible. 37, 10 minutes. Okay. And so we use reporter assays in these cells. And so the way this works is GLE here is driving a luciferase gene, and luciferase will take the substrate luciferin and emit light when it processes that substrate. And so by using a very low background camera, like a gel imager, we can actually start to see that if we overlay these things with cells that don't secrete hedgehog, we don't get any signal. But if we overlay these cells with cells that do secrete sonic hedgehog, then we get a very nice, robust light activation that we can quantify. And so we can actually get cost savings because we don't really need to add anything here. We've developed this protocol so we can actually just add the luciferin right in the media. We can pull this plate out, read it on a plate reader or on the gel camera and put it back in, read it the next day. There's no cost, it's easy, it takes 10 minutes. On a plate reader, it takes a little longer if we're going to image it on the thing. So it's very significant, very cheap to really run. The luciferase itself is really ridiculously cheap. What we also see is that if you have mesenchyme again that is responsive but don't have cells overlaid that secrete, you can add ligand back in and you can induce it. Ligand itself is really expensive to buy, and so the standard assays, this is what it looks like. You have to add in this amount of ligand to keep the signal up to identify any kind of an antagonist. When you have it in co-culture, though, you're automatically at this higher level emitting light constantly, and so you can dose response antagonize that without actually adding in that costly ligand. And so by doing that, you sort of save a lot of money as well. We can also start to see other important pathways, cellular responses that occur in response to this. So hedgehog induces proliferation. If we stay with K67, we see this gradient of proliferation near those overlaid epithelial cells and less proliferative cells deeper into the mesenchyme. We can also physically see the gradient that this is inducing. So even though all of these cells, including on this side, have this reporter, only these cells are secreting ligand. And so you can see that luminescent expression happens in this gradient formation, which is really cool because it really matches, even quantitatively, the gradient that you're starting to see if you stain this in a developing mouse. So this is the hedgehog gradient in a developing mouse, this is a hedgehog gradient in a plate. We can quantify these things and really play around with it. So it's a lot of cool, accessible biology. So really, we sort of have an ex vivo system almost. So if we want to look at this thing for screening, how does it compare? So we can screen with known antagonists of the pathways down here. These are all smoothened antagonists. But we also are very interested in identifying antagonists of other parts of the pathway, right? At toxicity testing, we don't really care how the mechanism of how something's screwing things up. We just want to know if it is screwing things up. And so if you add more endpoints that this thing's responsive to, you're actually getting a lot more value for your money. And so we can look at antagonists of hedgehog transcription, of cholesterol inhibition, of palmitylation, of hedgehog traversing through the ECM by binding to antibody, or by a specific Glee reporter. We see that in dose response, in every single one of these, we can generate nice dose response curves. So it's increased assay coverage. Here's the example. So these three are all in these parts of the pathway, 5, 5, 5, and 6. So these antagonists here. And basically the sensitivity is just as good, or maybe even sometimes better, than what we see in a 2D system. So we haven't changed our EC50 or lost any sensitivity. If anything, we've increased it. But we've also added, so you can see 2 and 3, that cholesterol or palmitylation inhibitor. If you do that in 2D, you get no response, right? Because they don't have that part of the pathway in the assay to actually antagonize. So you get no response there, whereas you see it in our model. So comparable sensitivity, but increased coverage. We can also start to develop new ways to sort of get more out of our model. And so one thing we found is that these things are really stable. They last a long time. And so we can grow them for weeks, and we've grown them for over a month. And they would grow longer. But basically what we found is we could do a dose response in these plates, wash out the signal. So we do a dose response, inhibit, and this was about 3 plates. We let those things recover for a week. Signal went back up, and we switched the doses around, high to low, different doses, different types, and generate the same curve. So now we have a lot more value here, right? We can do repeat dosing strategies and things like that to get more value out of each well. So one well is not just one chemical. You might be able to test 4 or 5, 6 chemicals per well, which is a huge, huge benefit in cost savings, and also developing new tox testing strategies. So in future directions, I've focused initially on hedgehog pathway, but as you can see, there are lots of pathways that are going to orofacial testing, and so we're working on those, but also going to broaden and see how we antagonize the other parts of the pathway. I'm also looking at chemical mixtures, so we know that we're not always exposed to one compound, And if one compound has a specific molecular binding to one part of the pathway, it could be binding similarly or to other factors in the pathway, right? If you have a specific interaction that's mimicked. And so we're starting to look at ways that we can look at mixtures and identify how these things might interact together. So I chose 2 pathways initially, and this is funded in my K99, to start to understand how these interactions work by testing targeted mixtures. So here I'm not screening for these mixtures initially, I'm testing targeted mixtures to understand if there are interactions that we might unveil that are yet to be known. We're also starting to look at, or want to look at gene-environment interactions to see if we can help basically prevent disease by identifying families that might be at an increased risk. So because we know we have this genetic and environmental sensitivity. So really, what have we built now? So with all of these strategies, I think we've really built a model that moves much more into the practical realm with things like repeat dosing, with things like luciferin in the media, to really decrease the cost and increase the throughput of these assays. Things that I'd say we can still improve on is, again, building more pathways into that model, looking at gene environment, and finally the main one is throughput that I think about a lot. And so we constructed this plate for the Transforming Tox Testing Challenge, where you have high throughput co-culture in a couple different wells. We show it works, and we've generated a decent amount of money, over $500,000 in funding to translate this into a commercial product, and we're continuing to develop that. In just showing that the model works, if you take an MCF7 reporter, dose it with estrogen, you have high activity all the time. If you dose it with testosterone, you have no activity all the time. If you put it in co-culture with hep G2 cells, which express a lot of aromatase, you get a nice dose response with testosterone, so it just shows that that functional co-culture works. We can also do that in this translational project. And so here we're basically in vitro trying to mimic what happens during different cycles of chemotherapy. So in chemotherapy, it's a very high toxic dose, and so you come in, you receive a dose of chemo, and you go home and recover for a couple weeks. You come back and you get a second cycle. And so our hypothesis is that we know in general that if you just take the cancer cells out and run that assay with chemo, it's not predictive of what's going to happen in the body. The hypothesis here is that the microenvironment is really critical in controlling or regulating how cancer cells respond to chemotherapy. And so we've taken patient samples isolated from tumors, and we've taken both fibroblasts and cancer cells. We do a dose response in different fibroblasts. So we have increasing doses, which is our cycle 1 dose that's only given to the fibroblasts in culture when this is not fluid connected. And there's basically little to no toxicity in the fibroblasts. But if you take those same fibroblasts then and put them in co-culture and give a second moderate dose of chemotherapeutic that's actually very biologically relevant, you can start to see with this patient here that so at low dose, if those fibroblasts saw little to no drug previously, your efficacy is high. You're killing, right? This is efficacious chemotherapy is killing. So you have a good level of cell death. If those fibroblasts had previously not seen any drug. And you see with this particular patient, you never have any protective value. If those fibroblasts, even if they've seen a high level of drug in cycle 1, during cycle 2 they're never really conferring much resistance. But in patient B you see a very different effect. And so drug here at low doses, yes, those fibroblasts aren't conferring resistance, but fibroblasts that have seen a higher level of drug basically now are able to totally remove the toxicity caused by this chemotherapeutic. So these are totally protecting this pathway in these cancer cells from death, which is really interesting. We see the same thing in this third patient. This is all with docetaxel. So we tried this again with a different chemotherapeutic that could also be clinically prescribed, mitoxantrone, and we see a very different effect. So in this particular patient, the stroma is conferring resistance to mitoxantrone. In this patient, mitoxantrone's having no effect, and in this patient we see actually a very interesting, where we see low-level sensitization, but then high-level resistance. And so here we have a patient that has mitoxantrone resistance, so clinically you would think you'd want to prescribe docetaxel here. you have mitoxantrone or docetaxel resistance, you prescribe mitoxantrone, and here you have dual, but maybe you can play with that dose and start to understand what mechanism is working there. Here the beauty is that you also have a functional assay. So even though we don't know why this is protected, we can start to screen with small molecules and identify potential known or unknown drugs that might target this pathway. So we've got really a discovery tool that even in a pathway we don't understand the mechanism, you can actually start to go identify druggable targets. You're at a much earlier stage or a much later stage in this sort of drug discovery process than you would be normally. So with that, this is, I think, so what do we do with our current models? I'd say, if I had to map them along this complexity relevancy pathway, I would say there's something like this. Where do they fit in the top paradigm? These models are very applicable for what I would call true high throughput screening, tens of thousands of chemicals a month. Here, things like hit validation or to test and inform in silico really explore biology. And also to test things like mixtures, gene-environment interactions, very specific hypotheses. So models are very useful, but for different contexts, right? So the complexity here that we've seen yields appropriate biology, and I think because if you target the biology that the clinicians, that the toxicologists are used to looking at, you're much more likely to build stakeholder buy-in and actually generate a risk assessment profile, because the end goal is that these things will inform how we regulate chemicals, which is $1 billion or $1 trillion problem. And so we want things that really give the right answer, not just an answer. And with the idea there that you might be able to skip animal models altogether, which would make lots of people happy. So that is the end. So really I started off with sort of the lumen model, which is really cool, but really branched that out through this micromilling approach to develop the orofacial clefting model, to develop this high throughput co-culture model. And from there, I've also been very collaborative and shared this device with people who are building things like neurovascular units, paraneural plexus, and collaborated and spread it out with other people at many other institutions. And so I think it's a broad, I think what this hopefully gets the point across is that it's a widely applicable set of technologies that have many uses. So practical solutions for 21st century topics. I showed data from Molly, from Ross. Pete's done a ton of work on everything. Tony's done in here. Jack mills lots of plates for us. And Dave for letting me do whatever I want. So there, I'll take any questions. 3 minutes over.